Hello again, it's Mr. McGowan here. This is part 2 of DNA replication. We're going to be learning about some of the material that we talked about already, the different enzymes, and we're going to then finish off talking about leading strands, lagging strands, continuous, discontinuous synthesis. And that's really what I want to stress, is what you need to know for this course. At this stage, the different enzymes of DNA replication, be able to label a replication fork, and tell me the difference between leading strands and lagging strands. You know I love repetition, so we are going to be repeating a few things over and over in this lecture as well. Helicase. That's the enzyme that divides up the hydrogen bonds, it splits it, it unzips the parent DNA into the two original sides. Now there's going to be a lot of tension that's released from the untwisting, and so the next molecule that comes in is topoisomerase, gyrase, and that relieves the pressure just so it doesn't coil back up again. And also to relieve pressure so it doesn't coil up again are single-stranded binding proteins. So in this diagram, these small yellow balls are supposed to be representing those single-stranded binding proteins, and they stop the hydrogen bonds from joining up again. That's called annealing. So they prevent them from re-annealing. Let's watch this excellent video.
Of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides, complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
The next major molecule we have to talk about is this enzyme, primase, or RNA primase, binds to the exposed single-stranded DNA and creates an RNA primer sequence of 10 bases long. So think of a zipper on a jacket. When you have the two sides of it unattached, you have to put a little starter piece into the zipper pull before the zipper pull will work. So that's kind of what the RNA primer is like. So it's a small segment that has to attach in order for DNA polymerase 3 to join on. It needs two sides to join on. It can't just attach onto a single strand. It has to join on to a double strand. So this gives it a little double strand to join on. So the sequence permits the binding of DNA polymerase to the strand, which begins elongation of the new strand. The DNA polymerase can only function in the 5' to 3' direction. So remember we talked about that there's two ends to a DNA molecule. It will add bases to the exposed 3', so the hydroxide end of the growing DNA strand. So it joins on to the 3' end. Since the parent DNA is made up of two antiparallel molecules, one strand will be elongated continuously and the other in smaller fragments. So one strand goes one direction, the other strand goes the other direction. They're antiparallel from each other. This diagram here is excellent at showing the parent strand. It forks, and then we have two strands. One of them will be made continuously towards the origin, that's called the leading strand, and the other one will be made discontinuously away. And it's actually the further pieces that are made first, and then these ones are joined on. So discontinuously. Again, parent strand. This is the gyrase. Here's helicase, which separates the hydrogen bonds. The single-stranded binding protein. First thing that gets laid down, this would be the discontinuous, this would be the continuous. So on the discontinuous and continuous, it doesn't matter. First thing that's laid down is an RNA primer, so 10 nucleotides long, made up of RNA. Once that is joined on, then you can have DNA polymerase 3. And for the continuous or leading strand, it will work its way towards the origin, towards the fork. There's discontinuous, which will work away from it. This is further along the strand. You can see the strand is actually being split this direction, continuously made through towards the replication fork, discontinuously being made away from the replication fork. This particular scene here is excellent because it shows DNA polymerase 3 from this Okazaki fragment is stopping. It bumps into primer that was already laid down from this Okazaki fragment. The DNA polymerase 3 will fall off. DNA polymerase number 1 will join on, and it actually replaces the RNA primer. So it removes the RNA nucleotide and puts on the proper DNA nucleotides. And a blow up of this, you can see discontinuous. Here's the DNA polymerase 3. There's your RNA primer. That's going to be removed by the DNA polymerase number 1. This scene also shows you DNA ligase. The ligase actually will join together those two Okazaki fragments. It glues them together. There will be a little gap, and it glues together those little gaps. DNA ligase. To summarize, always begins at the origin on the parent DNA. The gyrase will bind on, the helicase will bind on. It will separate them into two strands. It will have SSBs joining on to prevent them from annealing back together again. Primase will put on primer RNA, 10 of those, and that will be how the DNA polymerase 3 can join on. the DNA polymerase 3 joins at the primer and synthesizes, so it joins on the proper nucleotides. DNA polymerase 1 will bind to the RNA primer once the DNA polymerase 3 has come off, cut it out, replace it with the proper DNA nucleotides. Ligase will then stitch together or kind of glue together those Okazaki fragments to create one continuous strand. And DNA polymerase 1 and DNA polymerase 3, it's actually DNA polymerase 2, so we'll have to fix that one, can proofread the DNA, ensuring that the right base pairs are made to the parent strand. Remember, adenine with thymine, guanine with cytosine. Thank you.